today I have a pleasure to talk to you a little bit about Kids First program. We are kicking off now this community webinar. I will kick off because we have a really great program today, and I want to leave as much time as possible for our panel discussion. So this community webinar is in honor to Gabriella Miller. Gabriella Miller was a kid. She died when she was 10 years old of brain cancer. But Kids First is part of her advocacy legacy. Gabriella Miller in 2014 called the Congress and said, stop talking, start doing. And so this program, Gabriella Miller Kids First Pediatric Research Program, was authorized in 2014. And recently, Congress renewed the program just early this year in January for another 5 years. So we're really pleased with that. And today, we'll talk a little bit about who I am. I'm Marcia Fournier, the Working Group Coordinator and Program Director for Kids First. And the program is developing a data resource composed of multiple data types, genomics, clinical phenotypes, across pediatric diseases. And why is this important? Because with this data and the emphasis of the program of collaborative research and data sharing, this data can cross-pollinate across pediatric conditions and help researchers, clinicians, with discoveries of new tools for prevention, diagnostics, and therapeutics for pediatric conditions. Who we are. So we are trans NIH program supported by the Common Fund. So we work as a working group, as you heard. I'm the acting working group coordinator, and the working group is composed with NIH staff from multiple institutes. We collaborate with our partners, our researchers, the Kids First Data Resources Center, who you're going to be listening today from, the Kids First sequencing centers, who are responsible to generating the genomic data and other data types for Kids First, NIH staff, as I said, and you, the community, patients, families that are participating in cohorts, in research cohorts that are now part of the Kids First data resource. So Kids First, through this first 10 years, develop, as I said in the beginning, and is developing a data resource, and is one of the largest data repositories for pediatric data. So Kids First put kids first by having a dedicated data resource for pediatric data. So we hear from our researchers that how that is like make their life easy. They don't have to dig around which of the data is pediatric. All our data is really focused on pediatric data, pediatric conditions. So with that, you can explore multiple data types, learn about your condition, and more importantly, share that with your colleagues. And we are hoping actually to make these resources that now is heavily used by the research community to be also used by our communities, the communities that already participate as part of our cohorts. And this program, I think this is the fourth edition of the community webinar. The purpose of this program is to tell you how you can connect with Kids First program, how you can explore our data, and how we can learn about each other, and how you can participate on the program. So with that, I want to share one link so you can start exploring our data resources. You're going to see in the chat box a link to our portal dashboard where, without any login, you can start exploring the types of data, the conditions that we already have represented in the Kids First data resources. But as I said, this program is really emphasizing collaboration. We want to collaborate with you. Reach out to us if you don't see your condition as part yet of this resource. We would like to hear from you and learn how to work with you. And if your condition is already represented, also reach out to us if you have ways of expanding our resources or interacting and let us know how we can help you. Okay, with that, as I said, I don't want to take time from our panel speakers, and you're going to have an opportunity to interact with some of our community people that our partners, our research partners, the Data Resource Center, and NIH staff. So with that, I want to invite my colleague, Andréa Harris, who will be your master of ceremony today to kick off the panel.
Thank you so much, Marcia. Hello, everybody. It is wonderful to see you here. Please continue to engage with us in the chat. We are excited to see where you're coming from and to really sort of talk with you as we go. If something inspires you, type and tell us about it. If you have something, just a comment that you want to add to the conversation, please do send it. We're excited to see that and to engage with you here. I am Andréa Harris. I am a health science policy analyst at the NIH and a longtime part of the NIH working group leadership team for Kids First. By training, I'm a health services researcher and a social policy and public health expert. But I'm also a member of the rare disease community as a mom to a child who was born with and lost to short rib thoracic dysplasia, also known as June syndrome, which is a rare disease impacting about 5,000 people in the United States. I will be your moderator today, and I'm very excited to introduce today's panel. I'll start with Dr. Misha Mehta. She's a patient advocate and leader of the Neve Cult and Brave Ronil Foundation. Dr. Mehta was a rising leader in microbiology, molecular biology, and pharmaceuticals when her 5-year-old son, Neve, was diagnosed with DIPG, which is an aggressive form of brain cancer in 2020. His courageous battle and ultimate passing in 2021 propelled Misha into advocacy, where she's become a powerful voice for families navigating rare pediatric diseases. As president of the Neve Cult and Brave Ronil Foundation and a champion of investment access and reform, Misha is driven by one goal, which is to ensure no disease remains incurable. Welcome and thank you for being here. We also have Dr. Nara Sobrera. She is associate professor at Johns Hopkins University. Dr. Sobrera is a physician scientist specializing in rare scientific disorders. Her research focuses on identifying the genetic causes of rare conditions such as cartilage tumors and vascular anomalies, and applying those findings to improve patient care through targeted treatments and diagnostics. Welcome. We have Naomi Ojeda, clinical research coordinator at UC San Diego. Naomi is a clinical research coordinator for studies in pediatric neurogenetics. She manages the Gleason Labs International Spina Bifida Sequencing Consortium, leading patient recruitment, sample collection, and data coordination across various populations. With a background in behavioral neuroscience research, clinical psychology, and clinical outreach, she ensures rigorous family-centered research aimed at uncovering the genetic causes of childhood brain disorders. Welcome. And then finally, but definitely not least, we have Dr. Allison Heath. She's the Director of Data Technology and Innovation at Children's Hospital of Philadelphia. Dr. Heath leads the data innovation team at CHOP and is driving force behind the Kids First Data Resource Center. With a background in computer science and bioinformatics, she builds platforms that make pediatric data more accessible and usable for researchers, accelerating discovery across childhood diseases. Thank you all for being here. Welcome. To begin, I've read your bios. They're all incredibly impressive, but can you each tell us a little bit more about you and how you are connected with Kids First? We can start with Misha, if that's okay.
Thank you, Andrea, for that introduction and giving me an opportunity to share my experiences with the Children's Brain Tumor Network. Like you said, I got pulled into this with my son, Neve, being diagnosed with a rare pediatric brain tumor. My first interaction with CB10, and that's what I want to say because it's so powerful. They went above and beyond. They analyzed early enough my son's genomic data and all of those things that come with it, and identified potential therapeutic treatments that were not even out there in the clinical trial space. So for me, this community and the infrastructure that they're building holds a very special place.
Thank you very much. Dr. Sobrera?
Thank you for having me here today. I'm a physician scientist at Hopkins, and almost 15 years ago, when I started studying a very rare group of diseases, Kids First was one of the first places where I found support to investigate such a rare group of diseases. So that's how I connected with Kids First, was trying to find support to study this rare group of diseases, and Kids First was there for me. So that's how we started our connection with Kids First.
I'm the research coordinator for the Gleason Lab, where we also study pediatric brain diseases like Dr. Sobrera's lab. And also similar to Dr. Sobrera, we are connected to the Kids First as they are supporters of our work. And we are data contributors to the Kids First program, as well as we are users of the Data Resource Center. And with their help, we have made a few discoveries that we are proud of, and I will talk about a little bit more later. And we are also active, we have ongoing engagement as we continue to seek out their support and contribute.
As Andrea gave such a nice introduction, I got my start actually in grad school working on a lot of methods and algorithms, but in a lot of ways it was defined by having a lack of data. And every time you turned around to try and do something, it was very hard to get the data for the ideas or hypotheses that people had. So I've kind of been on this quest to build data infrastructure to support I think the next wave of folks trying to do that. Initially, I was at University of Chicago, where I led the build out of the NCI Genomic Data Commons, and I got the privilege of meeting Adam Resnick here at CHOP, who convinced me that it would be a good idea to come do this for pediatrics. And Kids First was one of the very first. The Data Resource Center was one of the first awards we got after I had moved to CHOP about 8 years ago. And it's just been a real privilege to be able to build out the technology and infrastructure that supports this amazing community.
One thing that really strikes me in hearing each of you speak and really resonates, I think, with some of the work that we do here is this idea of collaboration and the various perspectives that you each bring to the conversation. Can you each share a personal story or an instance in which collaboration has deeply impacted the work that you do?
I actually got to this group of diseases through a collaboration that was established when I was a graduate student at Hopkins. And just by having a conversation on the hallway with a researcher who was visiting from Duke, David Goldstein, he offered to do the sequencing. At the time, was the first whole genome sequencing that we were using in a family to identify a novel disease gene. And he offered, just from that conversation, to do that test in the family that I was investigating that day. And from that collaboration came one of the first genes being this gene disease association being discovered by the use of whole genome sequencing at the time in 2010. So collaborations are very at the center of everything that we do, because studying rare disease, we cannot do anything by ourselves. And the conversations can be anywhere among any people, can be a grad student and a PI, can be a patient and a physician. It can be not only among researchers. So we have to be talking and collaborating to get things moving and understanding better the rare diseases. So I think that was one of the first collaborations, and I was still a student, that showed me how strong collaborations and conversations and working together can be to solve patients, like to help patients to have a better answer for their health issues.
Misha, you've been instrumental in connecting researchers to valuable data, particularly DMG, DIPG. Can you share a story about introducing CBTN or Kids First data to a researcher who wasn't aware of it and what came from that introduction?
I also position myself as a self-appointed industry liaison. I want to like bring people together, like Dr. Nara was just talking before, collaboration, right? Like doing that. So in my self-appointed capacity, I attended ASCO, where I had a very interesting conversation that really struck with me. I met a researcher from a company that's doing fascinating work in like adult brain tumors. They've developed like quantum inspired AI platforms that can pull like clinically meaningful insights from like complex multiomics, even small challenging cohorts. And as they were describing their work, I couldn't just help but thinking that how this directly applies to DIPG, DMG, where the science is complicated, where the sample size is really small, and the need couldn't be greater, right? And they hadn't even considered pediatric application at that point. But as I shared more about what families like mine face and what incredible resources that we have in the Children's Brain Tumor Network, we have the curated data sets, biobank tissue, validated cell lines, right? The entire, consortium of things that you would need. You could see the wheels turning, right? And when I made the case for DIPG, that it's not just coming from an emotional plea, right? It is also a scientific, rich, data-ready opportunity. And what really clicked for them was, in addition of having all this data, is also to a way to enable them to validate their in silico works with those validated CBTN disease models, right? And that single conversation like turned into a real opportunity. I introduced them to the CBTN team, and now they're actively exploring like DIPG as a priority area. So it reminds me that an introduction to a data set is not just enough, right? You have to bring a story to it. You have to make the researchers see and feel the urgency and how they can even make like a meaningful impact in this space.
I think that connects very well to some of what you see on a regular basis, Allison. From the innovation side, can you tell us a little bit more about how Kids First Data Resource Center has impacted the connection among researchers, clinicians, and families? How you've made that bridge?
Yeah, I think it's been a amazing journey. I think, actually when you first said the collaboration, I think one of the unique things is at the very get-go, the Kids First and the Data Resource Center, we had an initial workshop where we brought in a lot of patients, advocates, families to talk through from the very beginning. And I think one of the things I often reflect on is, you have to, build out for the researchers probably first. We're talking increasingly about how does it become more clinical translation, but at the end of the day, like it has to come back to the patients and families themselves. And I think that's really, we've tried to build a base technology that will allow us to go in that direction and innovate in that direction. I will also say from the very beginning, I think bringing together the structural birth defects, congenital disorder community with the pediatric cancer community, I think has brought a lot of unique and interesting discoveries to bear. Just even seeing some of the scientific webinars where people present and they start riffing on a certain gene that they didn't realize was implicated in a different disease, and being able to bring people together to have more of that, Nara was talking about these kind of serendipitous interactions, but how do you accelerate that? How do you make it more likely to have a serendipitous interaction as we're trying to understand these complex diseases? That has been amazing. And then I think at the end of the day, always keeping the patient and families in the forefront, because that's why we're building all this technology. That's why we're doing all this research. I think as we think through it, and I think that's been a really unique part of Kids First, is even, the engineers and the cloud infrastructure folks, they try to stay connected to that mission as well. So all the way through all the different teams that work together to make this happen.
Naomi, in your work in neurological birth defects, Can you tell us a story where collaborative data sharing or cross-institutional work has sparked a breakthrough? I know you work in a different part of this timeline, right, and this process. Can you tell us a little bit more about what you've seen?
We have a very recent example of this in our recent publication in Nature on myelomeningocele. To provide a little bit of context, one of the conditions that we study at the Gleason lab is myelomeningocele, which is a severe form of spina bifida. And spina bifida is a neural tube defect that occurs very early in pregnancy, where the spinal cord fails to develop properly. And it can lead to lifelong challenges, such as mobility issues, bladder and bowel incontinence, and hydrocephalus. And it affects about 1 in every 1,000 births. So we started with a hypothesis that we thought de novo variants, which are variants that are present in the child and not the parents, rather than inherited, might contribute to disease risk. So this is something that required a very large sample size. And as this is rare disease research, we could not have done it alone, like come up with a big sample size. So we asked for help from various institutions, including patient advocacy groups like the National Spina Bifida Association, Rady's Children's Hospital in San Diego, other hospitals around the country and the world, clinicians, and even other universities who may have already been in contact with people with spina bifida. So that was, I can't even count how many groups that is, but in the end, we were able to assemble about 100 or 850 families for our cohort. But collaboration didn't really stop at collection, because we also did the sequencing, which is with the help of the Broad Institute at MIT and Harvard, and also with the help of other agencies like the Gabriella Miller Kids Foundation. We were able to generate whole exome and genome sequencing data. And through this large scale collaboration, we were able to identify 187 genes with potentially damaging variants, and uncovered networks involved in the closing of the neural tube, which is what spina bifida is the failed closure. So this would not have been possible without the decade-long effort of the group of people, which is now known as the Spina Bifida Sequencing Consortium, that brought together so many patients. And it's a reminder that in pediatric neurogenetics, especially for rare diseases, it's hard for a single center to do things alone. So truly collaboration is at the heart of our research, because especially, rare disease research. And currently we have a big problem that we still have some of the genes that we have discovered that are not recurring in our newer cohorts. So to kind of solve this, we just need to continue our joint efforts to get a better picture of how it is that genes are interacting, which genes are interacting, and how they lead to the development of diseases like spina bifida.
That's incredibly helpful as we start to sort of put it all together. We think oftentimes about transformative insights or the impact that we're looking to sort of all get to. And I think that it sounds like this is an opportunity to do that through data sharing. So Nada, you've applied data from Kids First directly to patient care. Can you tell us about a case where that access changed the outcome or direction of a patient?
I think one of the differential of the Kids First project, it's exactly that connection with the patient and that like commitment to get these results back to the patient. Many projects that the patients have participated before, they were enrolled and never heard back about anything, right? And there was no commitment to actually let them know any of the results of the research that was ongoing. With the Kids First is different, and I think each one of the investigators that are involved with the Kids First have that really close connection with the patients that are enrolled in the project. And that's not different in our case. And as I said, it's a very rare disease. We have enrolled now more than 300 individuals with this disease around the world in the project. And when we are analyzing it, we are always watching for what we can give back to the patient. What can we tell them about the analysis that we have done before the publication or independent of a publication? And that has been really like very eye-opening to me to have these connections with the patients and get the results somehow back to the patient. And in more than 30 cases, we have actually changed the diagnosis of the disease. For example, patient was enrolled thinking that they had one disease and turns out that they had a different disease. And it changed everything. It changed the risk of cancer. It changed the risk of recurrence in the family. It changed the way this patient is going to be managed and followed up for the rest of their lives. So, and in at least 5 patients, we identified a second disease. So this is very common with patients with rare disease. They come thinking that everything that's happening to them belongs to one diagnosis, and everybody's looking for one answer when actually they are like everybody else, entitled to have more than one disease. And so the secondary findings have been also something that have been very helpful for the patients to actually be aware of a secondary issue that they need to be watching for and taking care of. So that has been something that, as part of the Kids First, became even more part of our work. That is to work very closely with the patients to get the results back. Let them know if it's negative for now. Let them know when we find something that we are investigating further. Let them know when we find something that they can use clinically as the primary answer for the primary reason why they were enrolled, or as a secondary finding that we found. So we have actually had many examples of patients that we found a research variant, and then we worked with the patient to get that variant confirmed in a clear clinical laboratory, and then the patient has a report of the variant that we've identified initially in the research, and now they can use that for the clinical management, because that's important also. It's like something that sometimes the patients don't understand, that we are limited on the research side on giving results back, right? We cannot just give a result back to a patient from a research study because they cannot clinically use that for management. So we developed this path to work through the validation in a clear clinical lab so they can actually get a report and be able to use that for clinical management.
That's incredible that we're able to sort of build that connection from what we find from the research to the application in the clinic. That's a big deal. Misha, you're a strong advocate for open data sharing. Have you seen access to platforms like CBTN or Kids First help researchers move faster or uncover new insights, especially in underfunded areas like DMG?
I've seen how critical CBTN is in accelerating research, right? And like you ask in context of DMG, but I want to like say that it's not just like community like or investors that look in like drug development. They want to treat kids more than 400 that are diagnosed a year, right? However sad that sounds, but the beauty that I see about CBT, it's across the pediatric brain tumor. So if there are indications that are in multiple different types of pediatric brain tumor, and they can go after that one biomarker, right? So that kind of like then you're not treating one, you're not focusing on one group of disease, you're actually you're making a therapeutic development across the pediatric brain tumor spectrum, right? So for me, I feel CBTN has like really has been key in accelerating research and unlocking like newer insights, right? During my son's NEEV journey, I like often found out searching for options that did not even exist. I gave you my example of how I got introduced at the folks at CBTN early on. So that is the experience that drives my commitment to making sure that no other families, have to face these kind of limitations as they are traveling through these diagnostic odysseys. Not diagnostic odysseys, but this this journey. So one example that I think CBTN played a really pivotal role is in the DIPG DMG space is, like, I would say, the PNOC 003 precision medicine trial. It just not enabled, like, for rapid tissue and biospecimen or tumor collection, but it also supported the centralized processing and development of patient-derived cell lines, making all the data openly available, right? That's the key, making it open available. And that same data is now part of the open access resource and, like, open PBTA, helping researchers far beyond, like, the original clinical trial PIs and, the sites that were involved in to study DIPG, to develop, AI related tools to identify meaningful biomarkers. And it's this kind of open science that, gives me hope, right? Because in disease like DIPG, DMG, time is everything, right? And the open data gives us a chance to move forward together with a sense of urgency, right? So nobody's siloing the data. Nobody has, the first dibs on the data. And for me, that's the beauty of, like, having an infrastructure database like CBTN.
I love all of this. And I think it's really important to hear and continue to hear how we're advancing science, right? And how that then translates to the families. But I think there's also a role that Kids First and CBTN and other sort of large databases that are built in the way that we are can empower families through science, right? Can you all share a story that highlights how a family or a foundation really sort of close to you and on the ground in this battle has been empowered by access to data, participation in research, or a connection to the scientific process?
So the disease that I work with, they are mainly two as part of the Kids First, Olio disease and Maffucci syndrome. And as I said, when I started working with them back in 2009, very little was known about the clinical natural history of these diseases and about the genetic causes of these diseases. And the patients felt very lost. They didn't have a place to go. There was no community on the clinical field, on the research field, that understood and knew about these diseases and was interested to work with these patients. And the Kids First really gave this community a house. That's where they started to actually get organized as a group so they could actually move research forward for themselves, right? They felt like there was no place for them to get started. Nobody wanted to help them to know more about the disease. And when we started with the Kids First project, that all changed. And these patients are now organized in many different places in the world, but they're all connected through the Kids First project, and they are getting even more organized thanks to the Kids First project. So that was one example for me of how one project can make a huge difference for a rare disease. Like this one project really put that disease in the research map. And now, when I started, it was hard to me to find anybody to collaborate with because nobody knew about this disease and nobody was interested to learn more about it. And now I have a group of at least 20 people who are like researchers and clinicians who I work with in different projects, and at least 5 different projects, including projects that are now testing drugs for treatment of these patients. So in 10 years, the advance that we had in the research for this rare disease, thankful to this one project that we started back in 2017, it's huge. So that was one example for me.
Nisha, foundations often act as connectors. You've talked a little bit about your role as a connector. How have you seen patients and families and patient advocates successfully introduce Kids First or CBTN to data to researchers and their networks, and what can we learn from that?
So for me and in my community of parents, right, that we work with, like most of the powerful introduction that I've seen are not coming from scientists and industry leaders. They're coming from parents, right? Families who often stand up in the midst of their grief and saying, I want my child's story, even their tumor, to help the next child. that kind of courage kind of like moves this field forward, right? Because you're coming not only with data, like I said, but you're coming with a story that is impactful. So I've seen parents is like stand up on like stage at conference, speak up, not with medical jargon, right? But with clarity and a lot of heart. And I've seen me including like post on LinkedIn, tagging a researchers, even a biotech company, and saying that here's my child's story. And this is where you can access the CBTN data, right? If you want to study this is like a curated site. And these posts are very simple, but in my early example, right, they spark this new kind of curiosity that brings a new researcher into the fold, someone who hasn't even thought about it or even considered pediatric brain tumors, but didn't realize that this high quality harmonized data sets that is available now, and it's free for use, right, for the researchers community. And for me, it's like this is a truly catalytic moment, right? That is a shift in the researcher's perspective, that the data now does not even like, it's not just a sequence of numbers on a spreadsheet, right? It is a piece of child's life. It's part of their legacy. It's a gift from a family who's saying, please do something with this. And that kind of like connection for me, like just humanizes the science, and like it it feels like it fuels a purpose. It drives these researchers more than I've seen it firsthand, right? And what others can take from this is very simple but powerful. You don't need to be a PhD. You don't need to be a data scientist to, accelerate research. Sometimes all it takes is like knowing to point someone in the right direction, that and having that courage to do that, right? When we pair the right resource to the right, you know, person or the right institute, you're creating a ripple effect, right? And that ripple effect and then make converted to a wave, and then you're changing the tide of research in that little community that you're advocating for.
That's incredible. Allison, I'm gonna poke you here for a second. Tell us a little bit about how Kids First is building tools and access points to make data more usable and empowering for families and not just researchers. What can families do to get directly involved with Kids First?
I think sometimes when we point folks to specific tools or portals, it might seem a little bit overwhelming, but I think even just understanding the high level picture of, what is the genomic data we have. I've seen this Q&A question around, do you have health behaviors? But even just understanding, what are the different high level data points and being able to point people in the right direction, and then being able to, for the specific things that you might interest you, maybe going, the next level down, and having those discussions. And I think, engaging, I think what we found in the Kids First community, like, Nara and Naomi are saying, like the researchers here, the scientists here, they want to connect. They want to understand, what are those data types? How is that meaningful to individual stories? I think the questions you're asking, Andrea, are spot on. Like we all hold these kind of individual stories with us, and I think that is as powerful as whatever PhD data scientist who is trying to do an analysis. Those things are all important to connect. And I think the other thing that's occurring to me as I'm seeing some of the chat, I think the other thing that we've really tried to Kids First acknowledge, while yes, we take a lot of pride in being one the largest pediatric data resource, we have to integrate and be interoperable with other communities. I see a question about Down syndrome stem cell lines, and Wei Cheng answering about Include, which is another NIH program that we interact with very closely, and we have very good interoperability with, because we've in Kids First have sequenced some of the Down syndrome, but Include really starts focusing on all the clinical aspects, the different treatments. And I think that's one of the grand challenges of, we can't build a huge infrastructure for every single disease. So we have to come together and build some of that core infrastructure. But then how do we enable that ecosystem of, I'm not going to, our teams that work on the brain tumor side aren't going to know everything about, IBD, for example, or what disease you want to, but we have to be able to provide the framework and tools so that people who are in those disease domains feel so much more empowered by the data than feeling like, oh, we have to rebuild all this infrastructure just to get to the same point for the domain there. So I think that's a lot of what we think about. And where I'm going with that is then that gives the ability for families in the right disease communities to talk about the topics that matter to them, to feed into the researchers, and then the researchers can feed into what infrastructure, what data, what tools do we really need. And you really start seeing these amplifying effects when you start doing that.
Absolutely. And I think that that feeding of interest, that feeding of what families need to know, can definitely help to shape the questions that researchers are asking. Naomi, can you tell us a little bit about how, in your experience, you've been able to ensure that the experiences of the families are reflected and ultimately shape the questions that you're asking?
I agree completely with both Misha or Dr. Mehta and Dr. Heath. We view families as not just participants, they are partners in discovery. And I will share um, a current example of this at the end of what I'm about to say. But from the start, try to have our studies reflect what matters most to families by listening closely to their stories, challenges, hopes for the future, and more clinical aspects, like what it is that is affecting them. Um, Many families in our spina bifida research have shared deep frustration about the lack of answers, not just about why it happened, because most of them have no history of spina bifida in their family, and but also about what it might mean for their siblings or future pregnancies. So that the lack of answers pushed us to prioritize trio sequencing, and which is so just for context, we in our project we do sequencing on mother, child, and the father, because we are looking for variants that are not that are present in the child and not the parents. So we need all three. So this is why we're looking at variants that are not just inherited. We also, over the course of our research, we stay in touch with families, and not just through the consent forms, but through dialogue, newsletters, family webinars, and also ask for their feedback on how we communicate findings and how we design our study materials, because sometimes we need to know what is going on in their daily life to shape our questions. And then in the end, we want to inform them of our findings. I know in some studies, especially with more basic research, families are sometimes not informed, sometimes there's just nothing to inform, but we try to inform them of, even if we don't find anything, which is the case sometimes. Ultimately, we're asking scientific questions in part based on what is still unknown, uncertain, or misunderstood in our participating families. One current example is the PAX1 Foundation. I'm not sure if anyone is familiar with this condition, but the patients have variants in the PAX1 gene. And a few years ago, there was no foundation. And it was because of a patient family, a mother who started, like, she wanted more answers. At first, I think it took a while for her child to be diagnosed with this condition. And then eventually scientists were able to discover that her child had this variant, but there was no support out there. So she helped organize the PAX1 Foundation. And now there's many people, PAX1 families, that are involved, and research has come about from this. And now there's this specific mother has actually been on a co-author on some papers, because she is regularly consulted on if she thinks that the clinical questions are addressing the needs of PAX1 patients, because there's sometimes questions might be irrelevant, so she's there to help really fine tune the measures.
I'm going to shift our focus a little bit. We've talked quite a bit about the various parts of the collaboration and the empowerment along this journey. But I'd love to hear your opinions and thoughts on looking ahead. I'll start with Allison. As someone leading innovation at Kids First, what future initiatives or technologies are you most excited about, and how will they shape the next generation of research?
I think there's multiple fronts, and I think it also highlights maybe some of the multimodal aspects. On the genomic sequencing side, very excited about the rise of long reads and some of the new single cell sequencing and some of the new abilities that we have to really understand the genomic data. But in a lot of ways, I think it's how do we pair that with more ongoing longitudinal clinical data? So I think in a lot of ways, we have these time points where the genomic sequencing happens. We have what happened, to the patient at that point in time, but then follow up and continuing to follow up has been very difficult. And I think, as anyone knows who has tried to abstract clinical data and provide that, there's many challenges there. And so I have the fortune of being involved in a ARPA-H project called Radiant that I think is also a very close partner with Kids First, that we've been thinking through some of the challenges there. How does that actually ultimately feed into something like a Kids First infrastructure? And then I think I see this question in the Q&A as well of, I think part of why we're talking today in the community webinar is understanding where as we get these new discoveries, we have this new data generation, we're able to do quicker, more real-time analytics. Where's the first, foray of really starting to bring that back into the clinic? What time point is that? Is that in diagnosis? Is that in treatment decisions? Where can the new information, the ability to do rapid sequencing, the ability to bring in this clinical data, I think really excited to, think through, as folks are saying, kind of what Kids First 2.0 looks like. Now that we have this foundational research landscape, really excited, and I think really interested in how we, kind of do that revamp also of, times have changed since Kids First was first launched. How do we think through the next phase? And I think in a lot of ways, there's been a lot of great work through the Office of the National Coordinator in the US to allow healthcare interoperability that's enabling us to do new and interesting things that we weren't able to do when Kids First was launched 7 years ago, 10 years ago, I guess. And so now we really have this new opportunity to both increase the ability of data coming in and informing the research, but I think also the ability to figure out the right points to start, whether it's a pilot or initial way of, how does Kids First, from the data perspective, really start more directly impacting on the clinic side?
What kinds of collaborations between academia, families, and clinicians do we need more of in order to accelerate this discovery?
Well, I do think that the next step that excites me is actually the connection of all these data in different levels. So you see the Kids First data in a platform being shared with the public, with the community. And then, like that project, there are many others. And these projects need to be connected so we can actually accelerate the discoveries, because if we are talking about rare diseases, these connections and this putting data together, it's even more important. So I see as moving like a little faster on that side of the research that is actually connecting, is sharing the data, but connecting the data also. And I have been saying, I think we have now in the world enough data that if this data is actually all connected, we can make amazing discoveries much faster. So I'm much more excited as excited as I am about generating data, I am even more excited about connecting the data that's already out there. And this data is not only in the researchers' hands. This data is the clinical laboratories. This data is with the patients. The patients themselves have this data. And I think the hospitals, right, have data. I think we have to go beyond the data that's in the research labs and connect the data that is in different levels all around the world, in right now isolated. But I see a movement towards connecting this data in a more efficient way. And I think that will allow us so many more discoveries so much faster. So that's what I'm really excited about coming next. And I'm sure we will see amazing outcomes from connecting all this data. across different labs, clinical research, patients, hospitals.
Misha, as a scientist, a mom, and an advocate, what's your hope for how foundations and research institutions can continue to work together to make a difference for children facing rare disease?
So I'm going to piggyback on all the answers about, right? Like, and start with an example of what Allison was talking about, the Project Radiant. And for me, like, I'm like really excited and wanted to say that it's going to make a truly game changer is like to have that iteration from going from bench to bedside and back to bench, and then having that real time, right? We've been talking about clinical data. We've been talking research grade data. It has to be clinical data. And how does that transition happen? And it has to be in real time, right? Like for disease like my son had DIPG, we don't have that time. We don't even have an extra day, to speak of. So that real-time iteration of like applying and mapping a child's existing conditions or mutations across this landscape of data where there's multitude of kids there across disease types. What can we learn? How fast can we learn? And how do we incorporate into the clinical care? And I think we are getting there. We're not there yet, at least in our space, but I think we are getting there. Due to the efforts of like CBTN, the Project Radiant that's happening, that's going to facilitate all of that. And then something that Naomi said was like really true, right? Like, I feel that for me, collaboration is like not an optional. It has to be essential. And the foundations move with a lot of urgency. They're driven by personal experience, right? They're nimble. We are connected by families. We want to fill the gaps. We do that, right? In terms of the investment, the government invests about 8% in all pediatric brain tumors. The rest of the money, the bulk of the money is by family foundations doing a 5K or a bake sale. So we can hold our researchers accountable and saying that you have to share this data. Whatever we are funding, that data set has to go in part of a larger repository so everybody can learn from it and learn faster and quicker. And that is the partnership that you need. And I also feel that we need more parents, right? Like the example that Naomi gave, we need more parents. We see our kids day to day, 24/7. We have that lived experience. We know what is essential, how to design the next set of clinical trials. What are the outcomes that you truly should be looking at? It's evolving, it's slow, but like for me, the future is like, how do we dissolve these boundaries, where foundation are not just conveyors or funders, but they're true collaborators in clinical trial design and data strategy, even generating the right kind of hypothesis. That's the example Naomi gave, right? Like that is where I think the future is, and we're going there. It's starting to happen, right? But we just need to go faster. It gives me a lot of hope, but I just think we need to accelerate that process, and it should be across disease types.
That's really important. And so thank you for highlighting and sort of reiterating those points as well. Where do you see the biggest opportunity for new discovery if data sharing and collaboration continue to grow?
To reiterate, collaboration is at the heart of our research, as I mentioned earlier, connecting signals across borders, state, and maybe even international. So yes, no single hospital has enough cases to truly make meaningful discoveries. So, and right now, as I mentioned in our spina bifida research, we've sequenced around 1,500 parent-child trios, and we identified a chromosomal deletion that was a strong risk factor for spina bifida and was already associated with other conditions. Now doctors and families are able to look out for these additional conditions that they previously did not know. We know from other complex conditions like autism and congenital heart disease that it takes around 5,000 trios to consistently detect recurring risk genes and to start unraveling the modifier genes that shape how severely a child is affected or how they respond to interventions. So the path forward, I think, is data sharing. As it grows, we'll move beyond isolated findings and start to build a comprehensive map of risk, one that spans genetics, ancestry, environment, and outcome. We think that that's what will ultimately lead to new diagnostics, prevention strategies, and more personalized care for families. For us, now, and relating it back to the Kids First cohort, we actually just applied for a new grant to go even deeper and look for what are called mosaic variants. These are genetic changes that occur after fertilization and affect only some of a person's cells. So we want to know how these mosaic variants may affect structural birth defects. So we're excited about this because these are very hard to detect just by ourselves. These are very hard to detect with standard methods. But we believe that the missing explanation could be hidden in these mosaic variants that are only present in some cells, in cases where families don't have answers yet, so where no inherited or de novo mutation was found. So yeah, they could be the missing answers that families have been searching for. And we plan to use the advanced tools in the Kids First computing environment to reanalyze the thousands of samples that are already there across the different cohorts of structural birth defects, to look for these subtle mutations. So for us, the Kids First has been not just a data resource, but an engine of discovery and a model for what collaboration in pediatric research looks like.
I just want to take a moment to thank you all so much for sharing your perspectives, for sharing your wisdom, and for being here with us today. If I could give you a huge round of applause in a way that isn't awkward over a webinar, then I would. So please know, on behalf of the Kids First team, thank you so much for being here and for sharing a bit about your work and your stories and all that is important to you. Thank you. We're going to shift gears here and start inviting questions. Pradeep asked, I understand clinical data sharing is a key for advancing the road to curing rare diseases. In parallel, is there a need for sharing stories and journeys equally important as families walk through the various phases? And while he's sure that the answer is yes, how available are researchers to connect with families, to share and also learn from patient families?
As a clinician and a researcher, I don't think there is a platform where patients and researchers can directly connect that is widely used, but there are some that have already started that process. So we have the MyGene2 platform from University of Washington. They actually aim to connect patients among themselves and with researchers. Some platforms that we have developed here as in my lab that are widely used also for data sharing, like GeneMatcher. GeneMatcher allows the patients to participate also and enter their data and match with clinicians or researchers that may be in the database. So there are few tools that will allow participation of the patients and where the patients can connect with the researchers, but there are not many of them. And I think patients don't know enough about them. GeneMatcher has 20,000 users, but only 1,000 of them are patients. So I'm sure not a lot of patients know of the tools that are available for them. But it's much easier for the patients to connect with the clinicians. And then if these clinicians are researchers, great. But if they are not, then connecting to a researcher directly, it's a little bit more difficult because I think they don't really know. I think NORD is also another venue that helps the patients to connect with researchers directly. So there are a few things. I think patients need to have more access to what's out there so they know how to use it.
I'd like to add here that for our community, we have, and this is something that's tied in with the Children's Brain Tumor Network, we have Gift from a Child. And Gift from a Child is for postmortem tissue donation. So each tissue that is given to the researchers at CBTN, there's a story attached. It's coming directly from a child. Well, there is no formalized platform, but we try to go into communities like scientific conferences, and share these stories and saying that this is the data that is coming from these kids. Would it help to have a platform where if a researcher wants to go and look for a story? 100%, it is important, but and as you know, bigger foundations, I've seen this, and I participate myself as a parent advocate on those grants as well. So I'm bringing my personal story on to with the researchers for a funding that they're looking for. I think that that's more and more happening, but there's not a formalized process, and that's not a platform where every parent has a chance to come in and say and share their stories.
And I'll say also, you know, it was an experiment we did early on in Kids First, and I'm just not sure folks were ready for it 10 years ago as much. Of we had a kind of community feature, people could tell their story. And at that point in time, I mean, we were also a lot smaller of a resource kind of initially as well. But I think kind of to that point, revisiting it, trying not to, you know, if we can partner with, you know, platforms that Nara was discussing. But then I think there is always this challenge too of, you know, each disease, each domain, you know, has their own identity, the things they care most about, and having the right mix of platforms that are customized towards that versus kind of a general, like, you know, bulletin board of tell us your story and we'll try to match you. There's some right middle ground there. But I think what we just increasingly see is exactly what Misha was saying, that more and more those are being connected to the data, to even the specific samples and other things that are being provided. And I think that's been very important and valuable. And I think it inspires people to think along those lines more too.
Patient here who is interested, take a look at MyGene2. I just checked, they have now 6,274 patient profile. They were developed for that. So if you want to share your story, if you want to connect with other patients, and if you want maybe to make your story available for researchers, check MyGene2. That's I think a good venue for specifically for that.
Add a quick question, or add a quick point, and then I will leave Marcia to chime in as well. At Kids First, we welcome those stories. So if you are connected with Kids First, whether it's through a particular research study, or if you just want to tell the story of how Kids First, CBTN, or any of our connected resources have influenced your journey, we welcome that. And please feel free to use our email address. I've seen it floating through the chat, and we'll send it again. But we want to hear those stories, and we'd like to be able to sort of amplify the messaging for how this work has influenced your journey, and or how your journey is and continues to be meaningful for others who are traveling a similar path. So you know, that's an open invitation. Marcia, I know you were going to say something. I'm sorry to cut you off.
I think you said actually what I was going to say is inviting, I think, the community to reach out to Kids First, even though we don't have a formalized platform, as was mentioned. We love to hear from you. And also I want to say, few already connected with us through this event here. One way that Kids First is bringing the community together is hosting events like today. So we get to know you, you get to know a little bit about us, but really the goal is that you reach out to us and we can follow up and we can try to make that connections. I think a lot of our connections are, as everybody that is here today is part of this community, you a testimony that we have this very like personalized way of making this connection. So we would love to learn about your story, and then if we can help to connect you with a researcher, with a doctor, with a group, everybody here I think has brings your network to this community, and then that's the way we can grow and serve everybody the best way we can. So please feel invited and feel part of this community already by being part of this event today.
We have one more, slightly more technical question coming from Sarah Fowler. What type of computational models can help to show the complex connections among biological factors through critical stages of development? Allison, maybe one of the better folks to tell us a little bit about that. Your thoughts?
I was actually just thinking that, you know, the last 5 years of my life have really been about infrastructure and less about the computational models themselves. And I think it is a very complex question. I think one of the interesting things also about in the pediatric and developmental spaces, you have this time factor, right? That you're really trying to understand those different stages, and that can be very challenging, especially thinking about, it's not just the genome, right? You have the different expression levels, I think the different proteomics aspects that are happening, and all of that is a very challenging computational problem. And I think I think also, and I think the other thing that I think of a little bit, this is some of the new integration and models that people have done with the cancer data and the structural birth defect data, because in a lot of ways that's development in two wildly different contexts, but you have very abnormal growth patterns happening, and people trying to understand the different pathways that are happening there. And I was actually almost going to point it back to Nara or Naomi a little bit about kind of what the latest greatest they've used in their research towards this.
I think the use of RNA-seq and proteomics and metabolomics in different cell lines, right? Different tissue types and different stages of development is helping, but we still have not mapped it all. We are far from having mapped it all. So we need a lot of more work beyond using saliva, blood, and some skin biopsy to understand these things. And that is very important, but it's also difficult to get done. So we have to keep pushing to get these differences among tissues in different times of development mapped, so we can actually analyze them and map them back to the disease state. So I don't think we have right now the data to do that across all the tissues and all the organs that we need, but I think a lot of people are working to get more information that is tissue specific, time specific in the development, and that would be very helpful.
Please do not hesitate. Take note of the email address. We'd love to hear from you. We are very grateful for your presence and your participation today, and we're excited to see this next chapter as we continue the conversation between the community and Kids First.